Hi, welcome to the screencast on the heart. This slide here I include. This is the Chinese physician who is credited with being the first person to accurately describe the role of the heart in promoting circulation. I read about this dude in a book called Stiff, which is written by an author named Mary Roach. I know most of you are probably too busy to do fun reading, but should you ever find yourself with some time to kill and you want to read a little bit of lighthearted, funny, interesting science stuff, Mary Roach has a whole bunch of books and they're all fun. Stiff is all about cadavers, and she writes a little bit about the history of cadavers in anatomical study, which is where this gentleman comes in. But the book Stiff talks about how cadavers are used today in medical research, body farms, where they study how people decay for crime investigation techniques, and a whole bunch of other stuff. Mortuary science, you name it, if it has to do with dead bodies, it's in there, and she makes it funny. But we should just talk about the heart. This slide, not important. Just know where the heart is located and that it is enclosed in the pericardium. What is more important is that you understand the layers of the pericardium, which is on this slide here. And I don't have good close-up pictures for it. But there's 3 layers. On the outside is a fibrous sac. So this is sort of similar to maybe the dermis of your skin, or a joint cavity. So it's like dense, irregular connective tissue. Then deep to the fibrous pericardium is a traditional serous membrane with a parietal layer and a visceral layer. So the parietal pericardium lines the inside of the fibrous pericardium, and then the visceral pericardium lines the surface of the heart. So that when your heart beats in your chest, it moves around a bit, it kind of twists a little bit, and instead of your heart rubbing up against your lungs or something else, you have a layer of pericardium rubbing up against a layer of pericardium with some pericardial fluid in between them for lubrication, so that the heart doesn't damage itself or the organs that are next to it. The anatomy of the heart we are going to leave largely to lab, but I just thought we should review its basic function if you're listening to this before you go over the lab stuff. Right? So your heart pumps blood. It's got 4 chambers, 2 atria, so atrium here, and 2 ventricles, ventricle here, ventricle there. I think I need a thicker. There we go. Now when I draw, it'll be thicker and easier to see. So basically the way your heart works is that blood comes in the right side of the heart through those 2 blood vessels and then goes into that atrium and then out there to the lungs, and then blood comes back from the lungs. Let's just get a different color for that. And thicker. Right, here's blood coming back from the lungs, goes down into that ventricle, and then up here. So you've got your 2 atria and your 2 ventricles, which I just made a mess of, but I'm not going to restart the recording because these things take too long. All right, so next up is the anatomy of cardiac muscles. So you might remember when we did skeletal muscle, that skeletal muscle fibers are very long, multinucleate cells, cardiac and parallel. Cardiac muscle fibers, in contrast, are shorter and individual branching cells. So if you look here, here's a cell right here. It ends here and it ends there, and there should be an ending of it over here, but they don't show it. So all of your cardiac fibers are small cells with defined borders where there's 1 nucleus per cell. However, they do branch and split, as is shown right here, and they're interconnected with one another, so they form somewhat of a network. Those interconnections between the two cells are called intercalated discs. There are two important protein junctions that make up the intercalated discs. You probably remember them from Anatomy and Physiology 1. First are the desmosomes, which are strong mechanical junctions between the cells that keep your heart from tearing apart as it contracts. And then you have gap junctions here, which you may recall, as it says on the slide, allows ions to pass from one cell to the next. So what this means is that all of your cardiac fibers are connected to each other in an electrical network. So if you have some sort of electrical signal or ion flow, ions can flow into this cell, and then ions from this cell can flow into that cell, and then they can flow in this direction, and they can flow in that direction, and they can flow in that direction. So all of your cardiac muscle fibers communicate with each other electrically, and that allows your heart to act as one big contractile coordinated unit. So this brings us to the different kinds of cells that you find in the heart, or fibers: autorhythmic cells, conductive cells, and contractile cells. Autorhythmic cells, as it says right here, spontaneously depolarize, which means they generate their own action potentials without any input from the nervous system or any other sort of cell. Conductive cells you can basically think of as like axons in your nervous system that are just carrying action potentials from one part of the cell to another part of the cell. And then the contractile cells are similar in function to skeletal muscle cells, but they have a few anatomical differences that we just went over, and they also have some electrophysiology differences, which we'll go over. So we are going to go into the different action potentials that you see in autorhythmic cells and contractile cells. And we're not going to talk about the electrical activity in conductive cells, but we want to know that they're there. And I would assume that their electrical activity is very similar to the contractile cells because they're not autorhythmic. So first thing we need to understand before we get into the details of the action potential is to just briefly review what hopefully we all know about cells. And so I'm going to draw a cell. Here we go. That's almost a cell. We'll even give it a nucleus. So if you may recall, all of your cells have the sodium-potassium ATPase and a similar resting membrane potential. So every cell in your body should have elevated levels of sodium outside the cell because of the ATPase, and elevated levels of potassium inside the cell because of that sodium-potassium pump. The one thing that you probably don't know is that, generally speaking, the calcium concentration is higher outside of cells than inside of cells. So once we know this, and we remember that every cell in your body has a somewhat negative resting membrane potential, which means there are negative charges accumulated on the inside of the membrane, positive charges accumulated on the outside of the membrane, we can predict what's going to happen when we talk about these different channels up here. So looking at the channels, leak means that the channel is just always open, right? Like a leak in a boat. It's not letting a lot of sodium through, but sodium is always coming through it. Then you have these two voltage-gated channels, fast and slow. The fast and slow do not refer to how quickly ions are flowing, but how quickly these channels respond to changes in voltage. So we're going to get a change in the membrane potential that's caused by the leak channels. The calcium channels are going to open first because they are fast. Then the potassium channels are going to open. So then if we cast our minds back to action potentials in Anatomy and Physiology 1, which we covered when we did muscle cell action potential and neurons, if you have a lot of sodium outside the cell and you open a channel that lets sodium go wherever it wants to go, sodium is going to go into the cell. If positive charge enters the cell, then the membrane is going to become less negative or more positive. If we look at where calcium is, we can assume calcium is going to do the same thing. When a calcium channel opens, that is going to make the inside more positive. So you can think of it as a depolarizing current. And then potassium, because it's high inside the cell, if a channel opens for potassium that lets it go wherever it wants to go, it's going to go out of the cell. So all the same principles that affect muscle cells and neurons, skeletal muscle and neurons, are holding true here. It's just the channels function a little bit differently, so the action potential is going to have a different shape to it, but the basic phenomenon are all the same. So now when we look at the chart here, it is divided up into 3 time periods, which I didn't label here, right? But it's 1, 2, whoops, I did that wrong because I'm not paying attention. 1, 2, and 3. So normally in an in-person class, this would be a big 10-point essay question. For online, I don't want you to have to type as much for each question. So I'm going to ask you questions about the different time periods, and you need to be able to describe what's happening at each time period. So in time period 1, we call this the pacemaker potential or the pre-potential. What is happening here is that sodium is leaking in or entering the cell through the leak channels. That is causing this gradual depolarization, which is getting you closer to threshold. Then once you hit threshold, your fast-acting calcium channels are going to pop open. That allows calcium to flood into the cell, causing this spike of depolarization. Then the slower-acting potassium channels open. At the same time, the calcium channels are going to close, but we don't really worry about that. So your potassium channels open, potassium leaves the cell, and this causes repolarization. So for each time period, be able to describe the channel, which includes the name of the ion, say which way the ion is flowing into the cell or out of the cell, and then describe the effect that it's having on the membrane potential. So it's either pacemaker potential, depolarization, or repolarization. Now, when we get to contractile cells, we're going to notice that it's a lot of the same stuff, right? So you've got sodium channels, calcium channels, and potassium channels. They're all going to open in the same order. So when we did the autorhythmic cell, it was sodium, calcium, and potassium. When we do the contractile cell, it's going to be sodium, calcium, and potassium. And they're going to have the same effect on the membrane potential, right? You're going to start polarized, and then sodium and calcium are going to generate depolarizing currents, or they're going to depolarize the cell, and potassium is going to repolarize the cell. So all the phenomenon are still the same. The channels are just a little bit different. So in the contractile cells now, you have all voltage-gated channels. So we are assuming that a contractile cell is going to be brought to threshold by one of its neighbors through the gap junctions in those intercalated discs. So you have 3 voltage-gated channels that all feel the depolarizing effect from the neighboring cell. The sodium channels open first because they're fast, then the calcium channels and the potassium channels. The one thing I will point out about the calcium channels is that they are slow to open and slow to close. They're going to stay open for what is an extended period of time. So here's our graph. Same thing. We're going to have 3 different time periods, and you want to be able to describe the ion channel, the flow of the ion, and the effect on membrane potential at your 3 time periods. And so here's your first time period there in orange, and 2 in purple, and 3 in whatever kind of color, blue, green, blue that is. So during that orange time period, 1 is rapid depolarization caused by the opening of the fast-acting sodium channels. So sodium flows in, depolarizes the membrane. Then number 2 is the plateau phase. Your calcium channels open, allowing calcium in, but they stay open for an extended period of time. So the membrane remains depolarized for an extended period of time. Then during time period 3, your potassium channels are going to open. Now potassium is going to leave the cell, allowing the cell to repolarize and return to its resting membrane potential. So those are those 2. Try and keep them straight. Do whatever you have to do to remember all that, and there will be, a handful of questions on the exam. You might also see a question where I just show you the graph and ask you, is this autorhythmic or contractile? That's something else you want to be able to say. Now, when considering cardiac muscle cells, we do have to understand the special role that the calcium channels play, which makes your heart electrophysiology and heart contractions different than skeletal muscle contractions. So don't worry about this first bullet point here. It is interesting to remember that you still in cardiac muscle cells have, this is supposed to, SR means sarcoplasmic reticulum. So calcium gets released from intracellular stores, not in the cytoplasm, but in a subcompartment of the cell, to actually cause the contraction, as well as the influx of calcium through the channels, which affects the membrane potential. But the important part to remember is that you have this plateau here, which means you're going to have depolarization and elevated levels of calcium in the cytoplasm for an extended period of time. So when you look at here, a cardiac muscle cell contraction, you see that it is generating tension over like 200 milliseconds or so. Whereas here, this is a skeletal muscle contraction because it is not depolarized for as long, and the sarcoplasmic reticulum isn't releasing calcium for as long. The contraction is less than 100 milliseconds in a skeletal muscle. So in skeletal muscle, you get quick contractile twitches, and in cardiac muscle, you get an extended squeeze. And that squeeze is there because you want the heart to contract long enough to actually get blood to move. If it didn't squeeze for an extended period of time, it wouldn't be able to contract as far and squeeze out as much blood. We're going to leave this screencast here because I just wanted to introduce these terms, and we're going to do most of the heart next week.